ABO blade groups and RH factors. Early experiments with human blade transfusion often resulted in the days of the patient for unknown reasons. In this year, 1901, it was discovered that there were three blade types, that is A, B, and O, and that mixing blade from different types caused an immune response that resulted in clamping, and the reason for these days. Type AB is rare and was discovered later. ABO blade type, an individual red blood cells will contain proteins of type A or B, or both type A or type A and B, or neither. When you say blade type A, blade type B, blade type O, or blade type AB, we are referring to existence of uh, proteins, a kind of proteins. Okay. For example, when you say group blade A on a red blood cell, this is a red blood cell, a disc shape. On the surface of the red blood cell, you can consider these molecules. Consider these as a type of proteins, okay, on the surface of red blood cell. On group B, you can see this kind of structure, okay, diamond structure. This is a kind of protein, okay, on red blood cell. Let's call this kind A. Let's call this kind B. If there is a mixture of this kind of protein, A kind and B kind, let's call this red blood cell AB, because it contains on its surface A protein and B protein. If there is no such kind of protein on the surface of red blood cell, we call O. Okay, here you can see there is no such structure. Then related with the group types, there are making of antibodies, and so the antibody antigens are very common names. They are there. And what is antibodies? The chemical which our body uh, makes to fight against disease. That are also proteins. Antigens are also proteins. Okay. When you say A, group A or blade A, we are saying what? Antigen A. B, antigen B. Antibodies in plasma for group A is anti-B. That means they are different, so our body will make a chemical or protein which can fight against this kind. For B, it is A. For AB, there is no antibody. Okay, because there are the two proteins there, then the body will not taught us it is foreign material, because it is also already there. In this case, anti-A and anti-B will be made in the blade group O, because there is no protein in this surface, therefore everything is foreign for this blade surface. So A is foreign for it, B is foreign for it, therefore anti-A and anti-B will be made. Antigens, just for A, it is A. For B, it is B. That means the kind of protein exists in the surface, that is antigen A, antigen B. Then for AB, it is both are there, A and B antigens. For plate group, there is no antigen. There is no protein on the surface. The body produces antibodies that will attack any foreign type. This is natural process. Alleles of type, this one, capital I and A, and capital I and B are dominant over the type I. Okay, these are alleles. Allele this one, and this one. There are three alleles. Blade groups are multiple alleles because of having three alleles here. So considering the type of blade and the genotypes, then the antigen, antibodies, and which blade donate to which one and which one receive from which one, and the like. Let's see. If genotype is small I, then the blade type will be O. The antigen is there is no antigen. Antibodies was protein type A and B. So anti-A and anti-B. The O blade type donates to all kind of blades A, B, AB, and O because as there is no proteins on the red blade surface of the O blade, when it enters into the human body or the patient's body, it will not cause any fighting with the existing blade types. If it is A, the A blade will not recognize that blade because there is no recognizable protein on the surface of O. The same is true for if the blade, the receiver is B, if it is AB or O, there is no problem. From whom the O type receives only from O. If genotype is A, I capital IA and capital IA, that is homozygous, the type is A. The same is true as capital I is dominant to small I. If it is capital I and A with small I, the type will be A. So in this case, both are blade type A. The antigens are the protein A type. The antibodies are anti-B. And to whom they donate is for A and AB. And from which blade kinds they receive is from from A and O. And the B is similar with like that of A. In this case, AB, the genotype B is AB, capital IA and capital IB. The blade type will be AB. Antigens are A and B. The antibodies none. That is, as there are type of A protein and type of B protein, if someone receives any kind of blades, let's say A, let's say B, let's say AB, there is no fighting because as there is already A protein, B protein, there is no foreign to that body. So that is it. Then it donates to AB and it receives from all kinds because of this feature. Okay, there is no antibody production, so it can receive from all kinds, but it gives only to AB. If it gives to O, then as there is no protein on A surface, then it will make antibody that is RH factor or D antigen. There are different kinds of RH proteins or RH factors, but the common one are called D antigen. There are naming D, E, and etc., but the common one is D. The RH factor, the second most important blade group system after the ABO blade group system. There are different blade group systems you can understand from this statement. So the major one is ABO, the second one is RH. This was first discovered in rhesus monkeys. Its name is RH because of it was discovered first in these monkeys. Then finally it discovered it found this in human body. The RH factor is inherited independently from the ABO play group. They are not dependent each other. RH is independent from ABO. Genotypes for the RH factor are plus plus, plus minus, and minus minus. People who are plus plus or plus minus possesses the RHD antigen and tests as RH positive. The plus indicates the existence of this protein. The genotype, this one and this one, phenotypically, it will shows the RH because plus is dominant to the minus. People who are minus minus do not possess the RHD antigen and test as RH negative. Minus is recessive, therefore minus minus means there is no that protein. Let's say this black here on the red base surface, black square is for RH factor. This yellow triangle is for A antigen and The blue triangle is B antigen. When you say a person is AB plus, there is this protein, A kind of protein, there is also B kind of protein, and there is also an RH factor, so AB plus. If the person red blood cell contains the A antigen, A protein, and the RH factor, we call A plus. The same is true for B. B antigen is there, the blue color, then here is the RH factor, then B plus. O is like that. There is no any kind of antigen, ABO type of antigen or protein, then only RH factor we call O plus. AB minus is just there is no RH factor here on the surface. There is no black square. A minus there is no only A antigen or A protein is here and there is no RH factor. So A minus. In this case only B protein is found on the red blood surface and there is no RH factor, then B minus. On the O there is no black square here. That is, there is no RH factor, therefore O minus. The RH factor is important to determine in blood donations and transfusions. The genotype plus plus and plus minus, the phenotype will be RH plus RH plus. Then minus minus RH, the phenotype will be minus RH minus. The blood proteins in this case is there is what? Blood protein, kind of D in this case. Plus indicates the existence of this protein. Then in this case there is no protein. That is why it is called RH minus. Blood antibodies, in this case there is no antibody production because protein is there, therefore there is no foreign material for that one. Then in this case, this this RHD protein is foreign for the RH minus, therefore there will be anti-RHD production or antibody production. This one can donate to RH plus, and the minus one will donate to all kinds, RH plus or RH minus. Receiving from this one, the plus ones can receive from RH plus or RH minus, and the RH minus receive only from RH minus. If it will receive from RH plus, then there will be what? Antibody production, and then there will be problem. RH sensitization. One interesting medical scenario involves an RH negative mother who carries an RH positive baby. This is very important. The baby of an RH positive father and an RH negative mother can be plus minus genotype or minus minus genotype. If the baby is plus minus, the first pregnancy causes RH sensitization in the mother because she is exposed to foreign proteins and builds up antibodies against them. Future pregnancies can be increasingly difficult as the mother's antibodies attack the baby because she has antibody for that antigen. There are four possible scenarios of a couple, one couple. The first scenario is, let's say the male individual is RH positive, the female could be RH positive. The second scenario is the male can be RH plus and the female can be RH negative. And the third scenario is the male could be RH negative and the female RH plus. And the fourth one is the male could be RH negative and the female could be RH negative. When the RH sensitization will be there is when the male is RH plus and the female is RH negative. In this case, when you say male is RH plus, this is phenotypically. The genotype could be plus plus or plus minus. If the male is plus plus and generally the female is minus minus, then all the children will be plus minus. Okay. If let's say the first pregnancy will be plus from male, the next the minus from the mother, then the baby is plus minus. There is no other alternative in this process. All will be one. Genotypically plus minus and phenotypically all are Rh plus. Then there may be some problem. The other genotype, considering the male having this genotype plus minus and the mother is already minus minus, then if plus from male and minus from female, then the baby will be plus minus. In this case also the same is true, plus minus. If from the father it is negative and from the mother it is negative, then in this case, in these cases, the babies will be minus minus and there will be no problem in this case. Therefore, if the father is homozygous for the Rh plus, the probability of having the Rh sensitization is very high, and there should be what consulting medical experts. In this case also the problem is 50-50. Okay, if the baby is plus, then there will be problem. As the conditions are random and the possibility is there that the first pregnancy may be minus minus, then there will be no problem. The second pregnancy also may be minus minus, there also no problem. If there will be 4 children or 10 children, there may not be problem if there will be occurrence of this process. In another side, if the first pregnancy is plus minus, the second is also plus minus, the third is also like that, then there may be the problem from the heterozygous father of Rh plus. Consider this and just go to hospitals and check for your Rh factors with your fiancé. Consider this: if a baby inherits Rh plus blood from the father and the mother is Rh negative, problems can develop if the blood cells of mother and baby mix during birth. Uh, if there is no mixing of the blood, there may not be problem even though the baby is plus. Okay, that is good. In this case, mother is exposed to Rh antigens at the birth of her Rh plus baby in the first pregnancy. Through placenta, you can see that through placenta from the baby, the Rh plus antigens will be crossed to the mother's body. And mother's body makes anti-Rh plus, that is antibody making is there. These are the antibodies. The Y-like structure is antibody structure. Then possible subsequent pregnancies. Then antibody from the mother will cross the baby and then attacks the fetus. During the mother's next pregnancy, Rh antibodies can cross the placenta and endanger the fetus. Okay. Thanks for watching.